Good afternoon, everybody. Today we're going to start talking about how to control virus infections. Today and next Monday, we're going to start with vaccines today. And those of you that have taken the Moshewitz immunology course may remember that I gave a similar lecture in that course. Well, you probably don't remember, actually, but it will be different enough. And anyway, repetition is always good to reinforce. So, as you know, so far from what we've talked about in this course, we get infected with viruses. We have a great defense system that, for the most part, can protect us against them, even though they're able to undergo change, as you'll see in a couple of weeks, and remain one step ahead of us. So the real question is, viruses have been around for millions and millions of years. We haven't been around very much at all. Can we gain the upper hand? Can we help our natural defenses to prevent and control virus infections? And maybe even more interesting, could we ever remove a virus from the planet? Could we get rid of it? We'll talk a little bit about that at the end. So today, vaccines to prevent infection, and then Monday we'll talk about antivirals as a way of curing an infection once you get it. Vaccines and drugs can be very helpful in many ways, but as you may have already understood from this course, the virus-host interaction is pretty complicated, and it's evolved on its own without us intervening. So for us to think that we can do so is a bit odd, and it's daunting. There's always an effect of what we do, no matter if it's a vaccine intervention or a drug intervention. And we'll see some of that today and next time. And when we test vaccines and then put them out in the real world, for example, different things happen. Vaccines that work in the lab often fail in the field. Resistant mutants arise not only to vaccines but to antiviral drugs. Antiviral drugs, as you'll see next time, can have side effects that you never pick up in a clinical study. And then the last 2 points have nothing to do with science, but you can make the best vaccines or antivirals in the world, but if you can't get people to take them, they're of no use. So people don't want to take drugs or vaccines, and some societies can't pay for them or don't want to use them for a variety of reasons. So today and next time, there are two common threads that connect the two lectures. And the first is that any time we block viral replication, we impose a selection that the virus tries to get around. Doesn't try, it evolves to get around that. We get resistant mutants, we get vaccine escape mutants, and perhaps we get selection for increased virulence, but not always. And secondly, viruses are obligate intracellular parasites. So they depend on the host. So however you try and intervene, you're going to have some effect on the host because they are intimately tied together. And these are the risks of therapy with antiviral drugs. They're the side effects that can happen, and we'll talk about those next time. Vaccines are the best way to prevent virus infections. Look at this curve of life expectancy from 1900 to 2000. You can see that it steadily goes up. And there's a nice little dip here in 1918, which is what? Influenza. And then it goes back up again. It's steadily rising to nearly 80 years old. And this is for a number of reasons, but vaccines and antivirals and antibacterials are a large reason for that increase in life expectancy. So what do vaccines do? They mobilize the immune response to prevent infections. So they take advantage of memory, which we've talked a little bit about, in the adaptive response. Most of the vaccines that we use mobilize the adaptive response. And very importantly, they break the transmission cycle. They reduce the number of susceptible hosts. Every virus needs a certain number of these to spread. And if you reduce them below a certain number, which differs according to the virus can't spread anymore. So actually, if we all were immunized against acute viral infections, they wouldn't spread the way they do. But we don't all get immunized for reasons that are beyond the science. So vaccines give you a protective immune response, and they mimic a natural infection and provide memory. So here's a review of a primary infection and then a memory response. We're looking here on the y-axis at antibody or cellular responses in the form of T cells. So you get an infection, you have a primary immune response, can be antibodies or T cells, doesn't matter for the purposes of this discussion. The infection, if it's an acute infection, the infection is taken care of, the antibody or cellular response declines, and in this subsequent period, which can be days or years, you have protective immunity. Then at some point in the future, you have another infection with the same or a similar agent. It may be a mild infection. It may not cause any disease, but then you have a memory response. You can see it's more robust and it's more abrupt than the primary response. So this is what we depend upon when we use vaccines. The vaccine will provide an initial response, and then you will have memory. So what is immune memory? It is simply remembering the pathogen. And a great natural experiment in immune memory took place on the Faroe Islands, just north of the British Isles here. In the 1700s, it was quite isolated and didn't see much traffic from the mainland. It was an outbreak of measles in 1781. This was actually studied by an epidemiologist. And for the next 65 years, there was no measles on the island. Now, measles is a virus that requires a good number of susceptibles to spread. It needs about 300,000 people in a population, susceptible individuals to spread. So there weren't that number wasn't achieved on this small remote island. So there was no measles. 1846, there was another outbreak. None of the individuals who had survived the original epidemic were infected. They were protected because they had immune memory. Everyone else was susceptible. They were probably born in the intervening time. So that was a natural experiment showing immune memory. It's in all the textbooks. So immune memory lasts a long time. And I say here it's maintained without re-exposure to virus. This is because for many years, a lot of people thought that memory was maintained by reinfection. Maybe reinfection in the absence of symptoms. But that's not the case. You don't need to be reinfected. You keep the memory anyway.
Do you have any idea of the number of people who were exposed to both outbreaks?
I don't. It's probably known. I don't have the numbers, though. Why do you ask?
I asked because it's just a 63-time out. I was just interested in how many people they were looking at to see if they were infected or not.
Yeah. Well, if there is an outbreak, there had to at least be 300,000 or more for that to occur. I don't know the numbers, though.
Right,
Yeah.
It's a 63-year difference, so I'm just curious about how many people were actually exposed to the second outbreak.
Yeah, I don't know. It's a good question. Yeah. Yes?
If you don't need to be reinfected in order to maintain immune memory, then why, when you get vaccinated with clinically produced vaccines, do you need to get revaccinated on a regular basis?
Because most of the time when that happens, the vaccine isn't giving you a good memory response. And it's something we don't really understand very well, but there are determinants of a good primary response, but then there's separate determinants of memory. And the two aren't always going together. So the vaccine isn't good enough at making those memory cells that last a long time, whereas a natural infection is very good at that. And this is the case with flu especially, which is being debated a lot. And if you read the literature, you see no one really understands why one virus and not another makes good memory cells. But that's the reason. So immune memory, of course, has to do with antibody as well as cells. They're T and B cells, memory cells that remain. And as I said, we don't understand what makes good memory. But people are working on this. It's a very interesting area of research. These cells are present and they have a heightened ability to proliferate, so they make a very rapid response and a very robust one, as you saw. And the key here is that a vaccine does all of this without making you sick. That's the point, right? And that's the balance. To make a vaccine that doesn't make you sick yet gives you good memory. And the two don't always go together. For the most part, live or infectious vaccines are better at making memory because they more accurately mimic a natural infection. But there are exceptions for inactivated viruses as well. So vaccines are here to stay. Despite the anti-vaccine movement globally, they're part of our existence. And we immunize everything. We immunize children, adults, animals of all sorts. Your pets, you immunize. And because of this, we've gotten rid of a lot of childhood diseases. People live longer lives, as we've said, because they don't die in childhood of measles, for example. And for the first world, they're an important part of our public health, routine immunizations, but not the third world. So they have yet to spread to poor countries because they cost money. So the first vaccines were actually done many, many thousands of years ago by smart people who realized that when individuals got sick, they usually didn't get the same kind of disease again. So in fact, the Chinese practiced the what's called variolation. That is, they had smallpox back in the day, and they would take the skin lesions from someone with smallpox and inoculate other people with them to prevent them from getting disease. Now, it was smallpox virus, so many of the people got smallpox and died, but many of them were also protected. So it wasn't the perfect vaccine. And can you imagine the anti-vaccine sentiment back then? People were happy to live 30 years, I suppose. In the 1700s, Jenner, though, put this into practice, and he, of course, noticed that women who milked cows and got cowpox on their hands never got smallpox. So he said, I don't know what this is, because we don't know about viruses at this point, but maybe if I take some of these cowpox and put it in a person, they'll not get sick. So he did that, and that started immunization. And he did it with cowpox, and the descendant of that virus in some way is used as a smallpox vaccine nowadays. Pasteur made the first real vaccine in 1885. Of course, he didn't know that it was a virus, because we didn't get to viruses until almost 1900. But he prepared a vaccine that prevented rabies, and this was a very tortuous but successful vaccine, and he called it vaccine in honor of Jenner, who had worked with cows and cowpox, vacca. So the smallpox vaccine, by the way, we don't use anymore because smallpox has been eradicated globally, supposedly. But when I was a kid, it was routine to get smallpox. So got a little scar here because you would get an immunization with one of these bifurcated needles. They would put a drop of the vaccine in this needle and just scrape it on your skin just to inoculate the surface layers of cells with the virus. You'd get a little pus and then permanently scarred. Okay, I remember a kid walking around in the summer seeing people's arms always with the big smallpox scar. Some people had big ones and others had very little ones. But we don't do it anymore because smallpox is gone. Sorry? In the military, yeah, in case it's used as a bioterror weapon. But we're all now, a good fraction of the population is no longer immune and susceptible, and that's why we're stockpiling the vaccine and trying to make derivatives of it. That's right. After Pasteur, the first scientific vaccines was the yellow fever vaccine made in the 1930s, influenza vaccine followed, and then polio vaccine as well. Now, when you take these vaccines and you immunize people on a big scale, millions and millions of people, it works. So here on the top is the result of polio immunization. Polio in the U.S., 30, 40, 50,000 cases a year at its peak in the 50s. And the vaccines have eradicated polio from the U.S. And at the bottom, measles, again, high incidence in the U.S. The introduction of the vaccine got rid of that. So here are two ideas about how vaccines work and don't work. First is herd immunity, which is an often misunderstood concept. And all that means is that you immunize enough people to block the spread of the virus. All right? It doesn't mean that the virus is spreading to other people who are not getting immunized. It just means that you're reducing the number of susceptibles in the population to prevent the spread of the virus. So not everyone has to be immune. All right? That's what herd immunity is. And you also have to maintain a critical level of immunity for vaccines to work. If it falls below a level, epidemics will happen, even with good vaccines. Example, pandemic influenza. We have a vaccine, but the virus is still infecting people. You have to have enough people immune to block transmission in a population. Now, what constitutes enough people is complicated because it depends on the virus, the specific population, the environment, economics, infrastructure, social issues, all kinds of things come into play to this, quote, enough people. But for some viruses, we have some information, and here's some data on herd immunity. Again, the virus spread stops when the probability drops as a result of immunity. And this is virus and population specific. So smallpox, you need about 80 to 85% of the population immune to stop virus spread. But for measles, it's higher. It's a more infectious virus, more easily transmitted. You need 93 to 95% of the population immune as a result of vaccination. Now, the problem with these numbers is that no vaccine is 100% effective. So no, yes?
How do they infer the thresholds?
How do they infer it? A lot of mathematics that I don't understand. Maybe the guy who teaches the class before us would. It's statistics and epidemiology. It's looking at population data. You do vaccine trials and you do serology and you infer the serocoverage on the population, and then you look at infections. Yeah.
So they're determined before the vaccine is released to the public, not after it's already been released.
Yeah, you would have to do it at the beginning. Yeah. You can also use some naturally occurring outbreaks when you don't have a vaccine. If you just look at seroconversion in a population, you can get an idea of what the limits are. Yeah. So when you give a vaccine to 100 people, no vaccine will make 100 people immune to reinfection. That's a fact. So these numbers are a bit misleading. So for measles, if you immunize 80% of the population, if you give vaccine to 80%, only 76% becomes immune. That's the efficacy of the vaccine. So these numbers are even harder to reach than you think. So measles, for example, in the UK, when they stopped immunizing because of the autism scare, they went way below this 93%, and that's why there were outbreaks, despite having a lot of people immunized. So right now there's a mumps outbreak in the UK, and people are scared because a lot of people are immunized. Well, a lot of people may be immunized, but it's not enough to stop the spread of the virus, and that virus will find those susceptible individuals. The public plays a big role in whether vaccines work or not. So I collected a bunch of things that people say. I've heard over the years about vaccines and viral diseases. Viral diseases are a thing of the past. Polio is gone. I never get the flu. Why didn't you get the flu vaccine? Well, I never get the flu. Measles is just a trivial kid's disease. Chickenpox only infects kids. Kids should get infected naturally. I'm not injecting anything into my body. Vaccines make you sick. They cause autism. They cause MS. I know a guy who got the flu shot and then got the flu. That's a very good epidemiological study with N equals 1. I can't afford to immunize my kids. Oh, I don't have time this year. I told you that for many years about the flu shot. Although I did it this year. So I'm guilty of this one. I'm guilty of this one. So the societal approaches are a problem. You can have the best vaccine, but people just get in the way sometimes of using it. Public health policy depends on a lot of complicated variables. And I'm showing you this before we get into science because you may not appreciate that the perfect vaccine or antiviral is totally imperfect if all the conditions aren't right. Poverty, social structure, politics can affect the distribution. In developing countries, distribution is out of the question. They don't have the health infrastructure to distribute the vaccines. These large-scale programs where you immunize millions of people cost a lot of money. The government has to get involved. And of course, if economics are tight, it's a problem. And many people oppose large social programs. So they don't like to immunize large numbers of people at once. But it can happen, and a good example is the Gates Foundation, which has taken a big lead in donating money to immunize kids worldwide. They've given a lot of money to the polio eradication effort, and without money like this, they couldn't go forward because the governments that are the target, the countries that are the target, don't have the money to do this. So the dividing line, interestingly, between countries that can afford vaccines and can't is a gross national product of $500 a person. So that makes the difference. And most of them are located in sub-Saharan Africa. They simply can't afford to be immunized. It's a shame because we have vaccines for many diseases that affect these areas, but they can't be used there. But there are more problems than money. Many countries don't want Western ways imposed on them, and technology can run afoul of personal preferences and beliefs. For example, human fetal cells are used to grow viruses for certain vaccines. That bothers a lot of people because they're derived from abortions. And in 1994, an English schoolmaster banned the use of a measles rubella vaccine because, as he wrote, the products of abortion were used in its manufacture. The Pope actually did intervene, not the Pope, a cardinal did intervene in one of these cases and said, considering the benefit to society, or the potential benefit, it was okay to do this. And that guy who made that decision a long time ago is the current Pope, actually, who did that. So, how do you make a vaccine? So you have to make a good immune response. You have to make sure it's balanced between Th1 and Th2, which is not easy to do, and some vaccines have gone afoul of that problem. And the gold standard is a vaccinated individual must be protected against disease by a virus that would otherwise cause significant disease. Getting response is not enough. It's not enough just to make antibodies. They have to prevent infection. So there are two kinds of vaccines. We call them active or passive vaccines. The active vaccine is when you put a modified form of the pathogen or a piece of the pathogen into the host, and it induces immunity. And this is what we use for long-term protection. This is what mostly we're going to talk about today. But I want you to know about passive vaccines, where you put the products of the immune response into people. You put antibodies or immune cells into people. This is good for short-term protection, and a great example is rabies immune globulin. If you get bitten by a rabid animal or a rabid bat, you are often given rabies immune globulin before you get the vaccine so that you're fully protected. And the rabies immune globulin is derived from people who have antibodies. Here's a product sheet from one of the companies that makes, here are the brand names. So if you're ever in an emergency room and they're sticking this into you, you know what it is. It's used along with rabies vaccine to prevent infection. It works by giving your body the antibodies it needs to protect it against the virus. This is called passive protection. See, this is from the product data sheet. So I do tell you the right things in this course. The passive protection lasts long enough to protect your body until it can produce its own antibody. So it takes 2 weeks to get an antibody response. So in the meantime, this is protecting you. Okay. A passive vaccine happens naturally when children are born. They get antibodies from the mother. So this is these are antibody levels as a fraction of the adult level. And so here, these are IgGs that are passed from the mother across the placenta to the child. And here's the level at birth. And then it slowly declines. So the child is protected by all the repertoire of antibodies that the mother has during this time. And then the endogenous antibodies begin to rise. And this is why you don't want to give a vaccine to a newborn child, because it probably wouldn't work because they have so many antibodies transferred by the mother. You wait at least a couple of months. So with the rotavirus vaccine, it's tough because you have to give it before 3 or 4 months of age. So it's not easy. But other vaccines, you can wait a little bit longer. So what are the requirements? It has to be safe, obviously, don't cause disease, have minimal side effects, induce immunity in the population. And again, not everybody needs to be immunized. Doesn't have to be 100% effective. It depends on the virus, as we've said. That's where herd immunity plays a role. And it has to be long-lasting. Not every vaccine we've made is long-lasting. They don't have good memory, for example. And it has to be pretty cheap. WHO says it should be less than $1 a dose for it to be able to be used worldwide in all countries. It has to be genetically stable. It can't mutate. That's a big charge because, as you know, viruses mutate quite readily. How about storage? Many vaccines have to be frozen, and that's hard in remote areas. Interestingly, the WHO has developed kerosene-fired freezers for delivering the polio vaccine in remote areas, and that's worked pretty well. There's a new technology now that has just been developed where you can mix vaccine with sugar and desiccate it, and it remains very stable at even high temperatures. So this is very promising. And then the delivery issue is very interesting. It's best if you could drink the vaccine. A needle is tough because that requires trained health personnel and sterile technique and so forth. So if they're delivered by needles, it's another obstacle. Here are the different ways you can make vaccines. We'll talk about a few of these today, starting with our parental virus, which causes disease. We can make a live or infectious natural virus vaccine by a process of attenuation. We can inactivate this virus so it's no longer infectious. It's a killed or inactivated virus vaccine. We can break it up by detergents or chemicals, and this is a purified subunit vaccine. And then we can use recombinant DNA technology. We can clone individual genes of the virus. We can make live virus vectored vaccines. In other words, we can use a different virus to carry the appropriate antigen that we're interested in. We can make DNA vaccines. These are becoming very interesting. You take, again, the particular gene you're interested in, put that DNA into the host as naked DNA. And then finally, you can produce individual proteins of the virus and make either subunit vaccines, or in some cases, as you'll see, expressing, say, capsid proteins, you can make virus particles. So they're all different technologies. And these are some of the vaccines that are licensed and used in the US. And you can see that some of them are not used for everyone. So there are adenovirus vaccines, which are only used in the military. And you can see the different types. There are infectious attenuated vaccine, inactivated vaccines, virus-like particle vaccines, virus-like particles made in yeast or insect cells. We have the rotavirus reassortants. So this is what you can have. Here's our smallpox vaccine, which is certain lab workers or people in the military. So it's quite a range of vaccines, all made in different ways. So we're going to talk about some of these today. Let's start with inactivated vaccines. These are viruses that are inactivated chemically, and typical treatments include formalin treatment or propiolactone, or even detergents. The influenza vaccine is inactivated with formalin and a detergent. And that means that there's no infectivity. So this is a non-infectious vaccine. It's injected into you. And hopefully, during the process, the antigenicity isn't compromised. So again, it's a fine balance. You have to do a lot of experimentation to make sure that your inactivation does not destroy the ability of the vaccine to induce a protective immune response. So the first one we'll look at is the polio vaccine, the inactivated polio vaccine. And just to remind you that poliomyelitis is the disease that's caused by this virus, and that's where the name came from, the two Greek and Latin words. And this used to be a very common disease. This is a description from a textbook in 1959. As I said, in the US, 40 to 50,000 cases a year at its peak, and it targeted young children. So it was socially a very visible disease, big impetus to make a vaccine. Poliovirus, remember, is a small RNA virus with an icosahedral shell. And in the heyday of polio, the hospitals, as I've told you, were filled with iron lungs, helping people to breathe until their muscles could recover and they could breathe on their own. And the president of the US, FDR, had polio when he was older, and this really bothered him, so he decided to raise money to try and make a cure for the infection. So he started the National Foundation for Infantile Paralysis. It raised millions and millions of dollars, and the first vaccine that was produced by that research was the inactivated vaccine of Jonas Salk. It's treated with formalin, very simple. Take the virus, you produce it in cells, you treat it with formalin to inactivate it, and then you inject it. So there was a big clinical trial of this vaccine in 1954, still the biggest clinical trial ever done in the US. 1.8 million kids were involved. And this, look, it was 50% effective. So it protected half of the kids from getting polio. Not all of them, so it's much better nowadays, but for the time it was very good, so this was licensed and used. Now, to illustrate the kinds of things that can go wrong when you make a killed vaccine or an inactivated vaccine, well, here were the newspaper headlines that greeted this. This was a very visible disease. Everybody was happy when the vaccine was shown to work. But within a few weeks of release of this vaccine in 1954, polio began to appear in some of the kids who had gotten immunized. And this ended up being 260 vaccine-associated cases in 94 immunized kids and 166 of their contacts. So it spread from the immunized children to others, so obviously it involved infectious virus. And this was because one of the laboratories, Qatar, hadn't done the procedures properly for inactivation, and they released a virus with infectious poliovirus in it. They released a vaccine with infectious poliovirus in it. But eventually that problem was fixed, and the vaccine was used extensively, and in the 5-year period here, 55 to 60, the cases fell to about 2,500 in the US. So that was a very good vaccine. And if you're interested in this Qatar business, I recommend this book, The Qatar Incident, which describes the whole making of this vaccine and the problems. But maybe even more interestingly, this was the first time that a vaccine had failed in the public eyes. It was really the biggest vaccine that had ever been deployed. The public was dying for it, and when they got it, it caused problems. And all the vaccine litigation that we have today really originated with this vaccine. This book covers that really nicely. So poliovirus is acquired by ingestion, by fecal-oral contamination. It replicates in the mucosal surface of the intestine and then spreads below the epithelium into the lymph system and eventually the blood. And from there it may reach your central nervous system. Remember, the paralysis is a very minor effect. It's about 1% of infected individuals. What the inactivated vaccine does is it gives you antibodies in the blood that block infection. But you can see that in individuals immunized with this inactivated vaccine, the virus can still replicate in your intestinal tract, and you can still shed infectious virus. That vaccine, introduced in 55, brought polio down to very, very low levels in the US. Now, the other inactivated vaccine I want to tell you about is the influenza vaccine. And you recall that influenza is a different kind of virus. It's enveloped, although with an RNA genome, in segments. And for this virus, the main antigen that's important for protection is the hemagglutinin, one of the glycoproteins on the surface of the virion. The other surface protein, the neuraminidase, is less important, although it does play a role in protective immunity. So we have an inactivated influenza vaccine, which is available because there are a lot of deaths per year in the US as a consequence of influenza, and that's why we have this vaccine. It's grown in chicken eggs. It's inactivated with formalin and detergent, so the virions are basically disrupted, and you have mainly HA and NA present. We also have a cell culture produced vaccine that's been approved in the European Union. It's not yet available here in the US, and this is for people who have egg allergies. If you have egg allergies, you shouldn't use this vaccine grown in eggs. There are at least 100 million doses made a year in the US. Actually, most of the production is now overseas for the US. And it's about 60 to 90% effective in healthy children and adults less than 65 years old. There's some controversy over whether it is actually beneficial in older people or not. A lot of studies are ongoing to really address that, which is interesting because that's the main reason, one of the main reasons that the vaccine was deployed to everyone to protect against lethal disease in older people. And as I said, protection conferred by this vaccine correlates with antibodies in your blood to the hemagglutinin and the neuraminidase proteins. Okay. Now, the proteins of the virus change each year. It's called antigenic drift. And so we need to make new strains every few years, every year or two, to accommodate this. You have to make a new vaccine. And that's unusual, and it causes a lot of problems, as you can guess. To do this, we use what we call reassortants. We have a strain of the virus that's been used for many years, which grows very well in eggs. And you make a reassortment of that virus with whatever the current seasonal strain is. And what you want are the surface glycoproteins from the seasonal strain and all the other genes from this high-yielding egg-growing strain so that it grows very well in production conditions. And the vaccine for the fall in the US has been announced already. It has 3 strains in it, so it's a trivalent vaccine. It has last year's 2009 swine origin H1N1. That was the pandemic strain, which is now a seasonal strain. It has another type A strain, an H3N2 strain, which has been circulating down in the southern hemisphere. It's not clear that this is actually going to cause any more infections, but it's been put in the vaccine just to be sure. And then there's a strain of influenza B virus as well. So let's talk a little about subunit vaccines. These are ones where one approach is to break up the virus. The flu vaccine is sort of a subunit vaccine because when you break up the virus with formalin and detergents, what you're mainly getting is HA and NA. But sometimes you purify the components further. Another approach to making a subunit vaccine is you can take the gene. If it were the hemagglutinin of flu, you would take the hemagglutinin gene and express it somewhere and purify the protein and inject that into people. Okay. And typically you want to express a membrane protein like the HA of flu, or if it were a poliovirus, a capsid protein. And here are the pros and cons of a subunit vaccine. You make it by recombinant DNA technology, so you're never growing virus, so it's safer. It doesn't have viral genomes, they can't replicate, they can't escape. You don't have any contamination with infectious viruses, so that's why a subunit vaccine is good. The problem is it's typically more expensive, and the antigenicity is not great. It's just one protein. It's not an infection. The best immunity is conferred by an infection, so you can imagine giving one protein, it's difficult to approximate that. You get antibodies with these subunits, but often not T cells, and for some infections, those are important. And finally, they mostly have to be injected. And again, injection is hard. Not only people don't like injections, but as I said before, in some places, it's very difficult to do. And the common problem among subunit vaccines is the following. The viral proteins don't replicate, as I've told you. They don't stimulate inflammation like an infection does. Remember, when a virus infects you, it turns on the innate response. Innate response produces cytokines that cause inflammation, rubor, dolor, calor, tumor. And that inflammation is important for setting up a really good antibody response. And these subunit vaccines just don't do that. And just because they're single proteins, they don't have any nucleic acid, which is really good at tickling the innate response, other reasons as well. And here's a good example. Respiratory syncytial virus is a very serious pathogen, respiratory pathogen of young kids. And in the 60s, a vaccine was developed, which was a formalin inactivated vaccine, and it was tested in young children. But this failed, and it failed because it didn't induce a really good antibody response. We've just learned in the past year why this vaccine failed. Not only did it fail, but actually the kids who were immunized with it got worse disease than kids who were not immunized. And the reason was, by treating this virus with formalin, it destroyed its ability to be a good activator of the innate response. So the antibodies that were produced in those kids were not high affinity. And they bound the virus, but they didn't neutralize it, but they led to other more severe consequences. And so here's the paper that described this, was published in 2008. Lack of antibody affinity maturation due to poor toll-like receptor stimulation leads to enhanced respiratory syncytial virus disease. Great title, it tells you everything. So destroying this vaccine with formalin made it a really poor TLR ligand. So the sensor, one of the main sensors for this virus is a toll-like receptor, and the formalin inactivated vaccine didn't stimulate the TLR very well, so you got a very poor antibody maturation. Whereas the wild-type virus, the ligand is TLR4, get a great innate response and terrific antibody maturation. So any new vaccine has to take this into account. You have to make sure that whatever you do to the virus, you make sure it's still a good stimulator of TLR4. So we get over this problem of not causing inflammation in subunit vaccines by using adjuvants. All right? What is an adjuvant? It's a substance that stimulates early processes, and in particular, it makes a good inflammatory response. So where a subunit vaccine by itself doesn't cause good inflammation, you mix it with an adjuvant, and it will. And so it makes a better acquired response, and oftentimes with less antigen. And there are at least 3 ways that these adjuvants work. Through presenting antigens as particles, so the adjuvants are particulate and they present the antigen as a particle, which may lead to a better innate response. Localizing the antigen to the site of inoculation, it doesn't diffuse all over. Maybe this gives a better innate response. And as I said, it stimulates inflammation. In the US, we have adjuvants in some vaccines. The hepatitis B virus vaccine has an adjuvant which is composed of aluminum salts. Hepatitis B vaccine is a virus-like particle vaccine. It does not replicate, so it needs an adjuvant for a better immune response. And then one of the papillomavirus vaccines, Cervarix, has aluminum hydroxide and a lipid in it. It's an adjuvant called AS04. And those are the only 2 that I know about. In Europe, they use additional adjuvants in their vaccines. So this is an example of a good subunit vaccine, the hepatitis B virus subunit vaccine. And this is the capsid protein only, just one protein made in yeast. And that protein assembles in yeast into empty particles. And this is a very good vaccine. You don't actually need an adjuvant, but it's used to boost it and get more mileage out of the antigen. Because the particles that are made, these are the particles that are assembled when you make just the capsid protein in yeast, are particles. They're particulate, so they sort of resemble the virion, which is shown here on the left. So what we're expressing in yeast is just this surface antigen, and it makes incomplete particles. No genome is present. The other good subunit vaccines are the human papillomavirus vaccines, which are just licensed in the last couple of years. And there are 2 companies that make these, which are comprised of different serotypes of the papillomaviruses. One is made in yeast, and the other is made in insect cells, and this is the one with the adjuvant. And what's done here is to take one of the capsid proteins of the papillomavirus. These are icosahedral shells, right, formed by repeating subunits. They take one of the proteins, the L1 protein. They take the DNA encoding it, and they put it either in yeast or in an insect cell vector, and when it's expressed, it spontaneously assembles into virus particles. And these are then purified and injected, and they give rise to antibodies that block infection. So again, another example of a subunit vaccine. In this case, it's also a virus-like particle vaccine because the subunits assemble into virus particles. Let's talk a little bit about live, in quotes, attenuated vaccines. And in these vaccines, you actually get viral replication. So this most approximates a real infection and stimulates a good immune response. Depending on the vaccine, the virions may be limited to the site of inoculation. In other vaccines, they actually spread. The polio vaccine you ingest, and it spreads everywhere. And infection with these vaccines should induce inapparent disease or maybe mild disease at the worst. Now, when you have an inactivated virus vaccine, you typically have to give several doses because the virus is not replicating. So the initial dose is all that your immune system sees. It's not amplified. So you get an immune response, you give additional doses to boost the immune response, and get a good memory response. An infectious vaccine, you don't usually have to give many doses because it replicates. The red line is the course of virus replication. So the dose is amplified, and you don't have to give boosters. Sorry, the red line is the immune response. The blue squares are the virus response. So you can see the infectious vaccine obviously amplifies, and the killed does not. Now, how do you make an attenuated virus vaccine? Well, up until now, most of them have been made empirically by growing viruses in various cells or under different conditions until you get viruses that no longer cause disease. So you do some kind of growth. Here we're showing a virus isolated in human cells, and this is being grown in monkey cells. And the continuous growth in monkey cells selects for mutations that allow the virus to grow well in monkey cells. Then when you put them back into human cells, they don't grow as well and they don't cause disease. So now this virus has mutations in it which restrict its ability to cause disease. So this is an empirical process, trial and error. And this is what was done for the polio vaccines, the measles vaccines, and all the other infectious attenuated vaccine. So it's a very laborious process, and it's hit or miss. For the poliovirus vaccine, now, so this virus that we're going to talk about, the infectious polio vaccine, supplanted the use of the inactivated vaccine in the U.S. Many people felt that polio couldn't be eradicated without using a virus vaccine that approximated a real infection. So a number of people were working on these attenuated vaccines, and the first results were in 1952. Two investigators showed that if you pass poliovirus in cell culture, that virus would have reduced virulence in animals. So that was the first hint that this kind of vaccine might work. In fact, the yellow fever vaccine made in the 1930s is an infectious attenuated vaccine. It was actually the first one made against yellow fever virus. These strains of polio, the live strains or infectious strains, were licensed in the U.S. in 1961. And this is how they were made. This is the type 3 component of that vaccine. It's a trivalent vaccine. It has 3 serotypes of polio. And what Albert Sabin did, he took, for the type 3, an isolate of polio from a fatal case of polio in 1937, and he grew it in various hosts. You can see 21 passages in monkeys, monkey testicle cultures, monkey kidney cultures, on and on and on. And periodically, he would take a sample, maybe he would plaque purify it and put it into monkeys. He would inject it right into the spinal cord and brain until he found something that didn't paralyze the monkeys. Right? Complete empiricism. And he said he used 20,000 monkeys to do these experiments in the course of 10 years. But in the end, he got 3 vaccine strains that in clinical trials were shown to be safe and effective in people at inducing immunity, and those were licensed as the vaccine. So they were licensed in 1961, and in the 1980s, we acquired the ability to determine DNA sequence. And so the sequences of these strains that he had produced years earlier were determined and compared to the parent strains that he had derived them from. And these are the mutations in those vaccine strains that make them unable to cause paralysis in people. So here's the type 1, 2, and 3 strain. Now you can see there are very few changes. For the type 1, 5, and 2 each for the type 2 and the type 3 strains. So very few changes distinguish these vaccine strains from their parents, yet they've been used in hundreds of millions of people worldwide. Each of the 3 strains, by the way, has one mutation in common, which is in the 5' untranslated region of the viral RNA, in this highly structured region, which you may recall we talked about many weeks ago. It's called the iris. And these 3 vaccine strains have a mutation in one of these stem loops, stem loop number 5, which is shown here. And the type 1, the type 2, and the type 3 has all changes in this same region. And we're not sure how these work, but they probably just make the virus replicate a little more poorly, and that allows it to be an effective vaccine. So these vaccines are ingested. They're very easy to administer. You can just drink a little bit of the vaccine fluid, and that's it. You don't need any special trained health personnel. It goes to your mucosal surfaces of your digestive tract. It multiplies, sets up a viremia, and establishes good immunity. But it doesn't cause polio most of the time. It causes polio some of the time, but we'll get to that. So OPV, or oral poliovirus vaccine, protects your intestines from getting infected. It immunizes not only your intestines, but your blood as well. The IPV, remember, the inactivated vaccine just gives you blood antibodies. So this has an advantage, is that it prevents spread of the virus in the population. And that vaccine, the infectious vaccine, was introduced in 1962. It replaced the inactivated vaccine, and it led to the eradication of polio in the U.S. There's no more polio in the U.S. Last case caused by wild-type virus, 1979. But the vaccine does cause polio. It causes polio at a rate of about 1 case per 1.5 million doses of vaccine distributed. And that was first recognized in the 1950s, that the initial people who got this vaccine in the clinical trials excreted virus that was more virulent than what they had taken. And in the 1960s, there were paralytic cases associated with the administration. And nevertheless, it was believed that the use of these vaccines outweighed the possible side effects, so they were used extensively, and they continue to be used extensively. In the U.S., as we got rid of indigenous polio, that is the disease caused by a wild-type circulating virus, the only polio we found was vaccine-associated paralytic polio, which we call VAP, at about 7 to 8 cases a year, or 1 in 1.4 million doses. So we switched to the inactivated vaccine in 2000 because of this, because the only polio was caused by the vaccine in the U.S., so there was no longer any justification for using that virus. And this is what the polio looks like in the U.S. from about 1961. We're still just about stopping to use the killed vaccine. Introduction of the infectious vaccine, and we have a decline in the number of cases. And these, the line is the total cases of polio, and then the bars are vaccine-associated polio. So you can see a constant contribution of vaccine-associated polio to overall disease until eventually all the polio in the U.S. from after 1979, every case of polio in the U.S. was caused by the vaccine. So we switched to the killed vaccine and no more polio in the U.S. Now, why does this cause polio? Well, because there's only a few mutations in these vaccine strains that differentiate them from wild-type virus, and every time the genome replicates, there's a mutation sustained in it. So eventually, these mutations revert in your gut, and they may cause paralysis. And this is an experiment done many years ago by Phil Miner in the UK. He had a baby, and they gave the baby polio vaccine, and they collected the stools from the child every day or so for many weeks, and then sequenced the vaccine that the child excreted. So this is the type 3 component, which has a U at 472, and that slowly changes to C in replication in the baby's intestine. And the C makes this virus virulent and able to cause paralysis in monkeys. So this is why we get vaccine-associated polio, and it's one of the possible negatives of using an infectious vaccine. The other infectious vaccine I wanted to tell you about is for influenza. So we have a killed influenza vaccine. We also have a live attenuated nasally administered vaccine called FluMist. It has 3 different strains in it. These are actually reassortants. So a master strain was made years ago, which is cold sensitive and attenuated. And that's used to make a reassortment or a reassortant with the currently circulating strains. So we have the surface antigen that's relevant, and then all the other genes inside that confer attenuation. So these donor strains are cold adapted, temperature sensitive, and attenuated. So they have mutations conferring 3 different characters. Cold adapted means they replicate better in the upper tract, so they don't go down into your lung. Temperature sensitive, they don't like to grow in the lung as well. And then attenuated, they don't cause as much disease. So you get these squirted into your nose, they replicate and confer immunity. And this is how these are made. They take the donor virus, which was made in 1960, and it's cold sensitive, temperature sensitive, and attenuated in ferrets. They co-infect cells with whatever the current seasonal strain is, and then they select for reassortants that had all the genes from the donor with the exception of the HA and the NA genes from the current strain. And then they grow them up, purify them, and release it as vaccine. But what about the future? I think that one of the really neat ideas for a future flu vaccine is a virus-like particle. So it turns out that you can express just the viral hemagglutinin protein in a cell, nothing else, and you have virus particles made like this one. So these are lipid-encased particles. They don't have a genome in them. They just have a lipid envelope and the hemagglutinin in their surface. And these have been shown to be immunogenic and produce protective antibodies. So HA alone gives you particles. Now, this has been done in mammalian cells and insect cells, but what I find the most interesting is it's been done in plants. You can engineer plants to make these particles. And you can do it in tobacco plants. They're very easy to genetically engineer. We are really good at growing large masses of tobacco plants. Okay? So you can make a square meter of tobacco making these particles, it makes 20,000 doses, less than 20 cents a dose. Really, really cheap. And these are currently in clinical trial. My colleague Dick Despommier, who's here, he has developed this idea of a vertical farm. You know, we're running out of land, so we have to find a solution to growing crops. And his idea is to grow them in skyscrapers. Okay? So it's called the vertical farm, and this is his shtick, and you can see him here holding corn made in a vertical farm. But I think if you made this vaccine in Dick's vertical farm, it would really be very interesting. Anyway, so Dixon is going to come here in a couple of weeks and tell you about West Nile. Now, the neat thing about this approach is that you can do it very quickly. So here's a timeline for making a vaccine. So last year, when the pandemic strain was identified, a company in Canada called Medicago, they got the sequence and they cloned it into a vector and expressed it in plants, and they had a vaccine by the time the first wave of pandemic started globally. So about 4 months it took them to make a big batch of vaccine. The egg-based vaccine supply didn't start kicking in until 6 months later. So they were way ahead of the curve. And so maybe if we had lots of vaccine early on, we could have stopped a lot of the spread of this virus. So I think these plant-based vaccines have a lot of promise. They're cheap, and they're very quick to make. So we'll see how they work out. Yes? Nobody got it. The company was experimentally making it. They stuck it in ferrets and mice, and now it's in a clinical trial. Yes. Yeah. So can you eradicate a viral disease? Smallpox has been eradicated since 1978. It's the only disease that we've ever eradicated. And you need two things to be present in your virus or pathogen to eradicate it. You can only have one host. If you have multiple hosts, you can never eradicate a virus. And vaccination must induce lifelong immunity. So can someone tell me one virus that can never be eradicated? What? Look at these criteria. AIDS? Well, it probably could be because it's only in... HIV is just people. Well, there's SIV in monkeys. So the problem there is that if you eradicated HIV, maybe it would come back from monkeys again. You know, so that... But let's say it's the host range of HIV is just people, then it could be eradicated. But what virus is in many, many kinds of animals? Yeah. So flu is in birds, you know, of all kinds, seals, pigs, horses, dogs. Forget it. Never going to eradicate flu. But polio could be eradicated, as you'll see. Now, before we leave smallpox, there are two official stocks of smallpox in the world. There's one in Atlanta, and there's one in Russia. But there are other unofficial stocks, and we don't know where they are, and that's why we stockpile smallpox vaccine, because we're not sure who has these big batches of smallpox. Before Russia, in the Soviet Union era, they made a lot of smallpox as part of their weapons program, and we don't know where that ended up, and neither do they. So some people think that it could be used someday, and so that's why we're immunizing the military, because there are gallons of smallpox virus somewhere that could be used. And it's a pretty infectious virus. In 1947, when smallpox was under control in the U.S., a guy walked into New York City from Mexico, and he had smallpox, and he didn't know it. And he spread it to about a dozen people, and there was a huge, massive immunization campaign to contain it, which did work. But he could have potentially infected millions of people with a lot of fatality. So this is a good thing to have stockpiled vaccine against. So WHO wants to eradicate polio, and in 1988 they said we're going to do this by 2005, and eventually the goal is to stop immunizing because we're spending all this money and we want to use it on measles, which they want to eradicate next. So we haven't... Here we are in 2010, and there's still about 2,000 cases a year of polio in a handful of countries. Now, the problem with polio is that the vaccine can cause polio. Smallpox vaccine doesn't cause smallpox, ever, but polio vaccine causes polio. And we know that in 1 in 1.4 million recipients, it's causing polio. So eventually, if you get rid of polio and you're using OPV, which WHO is doing, the only source of polio will be the vaccine, as it was in the US up until 2000. So the WHO said because of this we have to stop using OPV, otherwise we keep having polio and it's caused by the vaccine, right? So you see the conundrum we're getting into here. The assumption was that the day you stop using OPV, the virus stops circulating and you don't have any problems. But that was shown to be false. Here's the progress of eradication. You can't see it very well, but in 1988 there were about 500,000 cases globally. And now in 2008 we have about 2,000 cases a year. Nigeria, India, Afghanistan, and Pakistan are the countries where it's never been eradicated globally. But there were outbreaks very recently in Egypt, DR, Haiti, Philippines, Madagascar, all caused by vaccine-derived polio. So these were in areas where immunization coverage dropped below that number needed to stop circulation. Polio outbreaks occurred, and they were all caused by vaccine-derived virus. Right? So these show you that the vaccine-derived strains can be virulent, which we knew, but they can also spread in a population. We also have immunodeficient people. They don't have B cells, and you don't know it for a number of years. They get polio vaccine, and they shed it. And they can shed it for 10 or 15 or 20 years, and they can spread the infection to others. So these are issues that tell you you can't just stop immunizing with OPV because it will continue to spread. Yes?
When people who are not immunocompromised get OPV, how long do they get polio for?
A few weeks. Maybe a few months at the most, but no more than that. Yeah. So there is also shedding by them, but they presumably would stop. The problem is that these other individuals could shed it for years. So if you stop using OPV, you have increasing numbers of non-immune individuals, and eventually this circulating vaccine virus will infect them and cause polio. So this is the problem. You can't stop immunizing because now you're immunizing against the vaccine, which is a crazy situation, but that's the way it is. So what's going to happen is when polio is eradicated, we will switch back globally to the inactivated vaccine, and then at some point where we say there's no more polio in the environment, we can then stop immunizing. But it's not a straightforward issue. You see, so having a killed vaccine is difficult because if you don't completely inactivate it, it can cause problems. And an infectious vaccine like polio that is very similar to the virus that causes disease is a great tool for eradicating a virus, but then you have the issue that it can cause disease afterwards. So this is a great example of the complexities involved in immunization. And after we do eradicate polio, which probably will happen someday, we have to keep stockpiles of the vaccine, just like we keep stockpiles of smallpox vaccine, because someone can use it for terrorism. There's also virus around, and we don't know exactly where it is, in all kinds of samples that are stored. The terrorism would come from having the sequence of the genome. You can easily synthesize this genome and make infectious virus. So you can never get rid of all the possible sources. You have to assume that an outbreak would occur, so you have to stockpile vaccine. So it's not like you can forget about it when the disease is gone. I want to leave you with this. Even if we can eradicate a virus, as long as we have the nucleotide sequence, we can make it. Even for a virus like smallpox, which is 250,000 base pairs, that sequence is everywhere. You can go on your computer and look it up and get the whole thing downloaded to your hard drive, and you could make the virus from it. It can be done. So I don't think we can ever eradicate a virus from the earth.